Hi everyone. So in this video today I am going to start with cell division. Cell division. What is meant by term cell division? See, there was a scientist called Rudolf Virchow. Rudolf Virchow. Rudolf Virchow stated that a cell always arises from a pre-existing cell. What he said? A cell always arises from a pre-existing cell. A cell cannot be made. You cannot make a cell in laboratory. You cannot mix all the material required or the Panchtatva which we have studied. Okay, these things, of course, we can come to know. We know that the cell is made up of protein, carbohydrate, fat, vitamins, mineral, DNA, RNA, and all. Even if we know the exact quantity of them, we can mix together and put it or blend it or whatever you do. You can never make a cell in laboratory. You cannot make a cell in any way. A cell will always arise from a pre-existing cell, from a living cell, biologically. Okay, this is very clear. There shouldn't be any doubt. A cell will always come from a pre-existing cell. Now, the cell can be of plant cell can be of animal cell can be of prokaryotic cell, or cell can be a eukaryotic cell, whatever it is. But cell will always come from a pre-existing cell. Okay, so this is one thing. Now, we must know what is cell cycle. Cell cycle is the cyclic changes which is taking place from one cell to formation of another cell. Like suppose we got a cell here. Just for the example, you just consider this is a cell. Now this cell will form another cell. So all this cyclic change which you are seeing here, this is known as cell cycle. Just I can draw the diagram circle, a perfect circle also, but why to waste time? So this is the cell. Now this can be divisible into two clear phase. One is this one from here to here. This is called interphase. 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 Interphase is that phase in which a cell is carrying out its normal activity. Every cell is meant to carry out certain activity. Like if it is a muscle cell, its function is to contraction and relaxation. If it is a bone cell, its function is to make secrete or make the bone stronger. If it is a retina cell, eye cell, rod cells, then their function is to see. If it is skin cell, their function is to protect. So there are different cells which are found in our body, and every cell has got some function. Okay, so every cell has got some function, and those functions which are carried out by a cell that particular time or that particular phase is called interphase. Cell is further divisible. The interphase is further divisible into three phases. This one is called G1 phase. This is called S phase, and this is called G2 phase. We shall study one by one what are the changes taking place in this. First is G1 phase. G1 phase is the phase, it is G1, okay? G is standing for growth. So it is a phase in which a cell is preparing itself to undergo under S phase. Okay. Now what actually happens in the S phase? And S phase is synthesis phase in which the new DNA is synthesized or new genetic material is added. So for that, some substances are required. What are the substances? That is, the DNA is required. So new DNA, proteins are required, other raw materials are required, and energy is required. So G1 phase is the phase in which a cell synthesizes new DNA, new protein, collects raw material, and stores energy so that these can be used in S phase and new DNA can be made, and this DNA will be used for synthesis of sister chromatids. So I think G1 phase is clear. G1 phase is the first phase in which a cell is preparing new DNA, proteins, raw materials, and energy so that it can be used in S phase. What are S phase? Let us see what happens in the S phase. Now, this is S phase, where S stands for synthesis. Synthesis means to make something. What is made here? See, a normal chromosome or chromatin material is appearing like this. Okay, this is one chromosome, and its homologous chromosome is like this. Homologous chromosome is the chromosome which is actually coming from other parent. This will come from one parent and that comes from another parent. They are exactly same in size, shape, position of centromere. Its centromere is here, so its centromere is also here. Length is same, bending pattern is same, everything is same. Even the genes which are found, suppose here it is the eye color gene, so here also it will be eye color gene. Here it is hair's nature, that is whether it is curly or straight, so here also it will be same. These are the chromosomes, these are called homologous chromosomes. Actually, there is no need to understand homologous chromosome right now, but just to clear have a clarity between the homologous chromosome and sister chromosomes or chromatid, I drew that. So I can just rub it off. इसका कोई काम अभी हम है नहीं हमारे पास, तो हम इसको अलविदा करते हैं, वापस आते हैं. So in the S phase, the new DNA are made and new proteins are made so that another sister chromatids can be formed. So these two are known as sister chromatids. The two sister chromatids, where are they formed? They are formed in the S phase of interphase of a cell cycle. G1 phase was only collection of raw material. Once the raw material is collected, no problem, it can be used or it cannot be used. But once the S phase is done, every chromosome will make its exact copy, and that copy is called sister chromatid. And the sister chromatids are found attached at centromere. As long as they are found attached with the sister with at the centromere, they are called one single entity. We will be calling it one chromosome. Though now it has two chromatids, but we'll be calling it one single chromosome. Okay, it is something like that. Our father was having a shop. Now the son is also become old, and he also started sitting in the sun. So the father may put a name, Ram Prakash and Sons. Okay, but the shop is one son or two son or five son, whatever. Now all the children, when they become little big and they get married, they may start out their own ventures. They may also put the name Ram Prakash and Son. But as the shop is separate, now that will be a separate entity. As long as both all the children were sitting in one shop, it was one entity. The same way, as long as these two sister chromatids are found attached at the centromere, it will be considered one chromosome only. If we separate them, both sister chromatids will be considered as two independent chromosomes. So that you must remember. So actually in S phase what happens? The DNA content is doubled. DNA content is doubled. And we know DNA and protein collectively together makes chromosomes. Okay? But as long as they are attached, they will be called same chromosome. Only the DNA content is doubled, but the chromosomes are same in number. Okay, that you must remember. The question comes in this way, in multiple choice. In S phase, a cell had 40 chromosome or 20 chromosome. After cell S phase, how many chromosome it will have? So you will have to write it as if it was 40, it was 40. If it was 40, it is 40 only. If it was 20, it was 20 only. The number of chromosomes are not increased. What is increased is DNA content. Okay. So this is a change as long as S phase did not take place. After G1, even a cell can decide, okay, I will not undergo cell division. Okay. A cell had a freedom to choose whether it had to go to undergo a cell division or not. But once S phase is come and the cell has divided its content, new DNA is formed. After that, a cell cannot go back or give up the plan of cell division. So after G1 phase, if a cell gives up a plan, okay, I will not undergo. This is called G0 phase. G0 is a phase in which a cell may give up or it may nullify the effect of cell division. But once it enters into the S phase, it has to undergo a cell division. This is very clear. One must remember very clear that once the S phase is come, a cell cannot give up its idea of cell division. It will have to undergo cell division. S phase is clear, G1 is phase is clear, G0 is phase is clear. G1 in which the cell is prepared for S1. But it is not necessary that after G1 a cell has to undergo S phase. It can even give up its idea of cell division and it can carry out its normal activity. S phase, the DNA content is doubled. Each chromosome may develop its sister chromatid. Now it has to undergo a cell division. So next comes G2 phase. What is G2 phase? G2 phase, now since the DNA content is doubled, it has to undergo cell division. So now remaining rest of the preparation it will carry out for cell division. But what it will do, it will store energy. Second, make new cell organelle. Increase cytoplasm. The volume or the content of the cytoplasm will also increase, so that when a cell divide into two, they will have sufficient amount of cytoplasm, sufficient amount of cell organelle, mitochondria, Golgi body, राइबोसोम, सेंट्रोसोम. एक सेल में जितना है, टू सेल के अंदर, दो सेल के अंदर उसके डबल हो जाने चाहिए, so that each cell will have the same, and some they will form later on. It's not that exactly same number of chromosomes, same number of mitochondria which is there in one cell will be there in the daughter cell. No, they will be initially less, but sooner or later they will be maintained or as per requirement. But initially their number will be, but in this stage its number will be more than the beginnings of the cell. Okay, so G1, S phase, and G2 phase, I think these three phases are clear to you. And the fourth one, this fourth block, this is for M phase. M phase is the phase of division, where a cell is to divide. Now, in fact, we can compare this entire process. Like a couple is there, a man is there who got married. After marriage, he has got children. Say, for example, he has got two children, two sons, or son or daughter, whatever it is. Now, in the course of his livelihood, his service, his working, he will collect things, earn money, save them, buy a house, buy some things, car or free washing machine, whatever things are required at home. Same time, he will be giving education to both the children. Once the children get married, now they can't stay in the same house, so he has to divide his property. So the both the children, the property will be divided. One house, the parental house. Another house will be given for the new child, the one of the son. Some material are here in this house. Same amount of material, same type of things will be in the other house. So this is how the two children will get the same type of thing. Here also the two cells which are there. One was the parental cell and one new synthesized cell. Both the cell will have same type of content. And interestingly, if you remember, in S phase, when we said the DNA is doubled, DNA is actually replicated. And when the DNA replicate, it's all the properties are exactly same. So a DNA, whatever type of gene was there in the parental DNA, same type of DNA will be there in the in the sister chromatid. So I think cell cycle is clear to you. Cell cycle is a phase in which a cell starts its journey from one cell and it undergo in interphase and M phase, and after the M phase it results the formation of another cell. One cell it was already having, another cell also comes. Cell cycle is divisible into four parts, or rather two phases, that is interphase and M phase. Interphase is divisible into three, that is G1, S and G2, and M phase is the next part. Now G1, the raw materials are collected and new DNA is synthesized, new proteins are made, and during this phase a cell is preparing itself to undergo a cell division. But it may also give up the idea of cell division. So if it gives up the idea of cell division, this will be called as G0 phase. If it continues, then it will enter into S phase. As it enters into the S phase, whatever chromosomes it has, the chromosome will have DNA and the DNA will be replicated and new chromatids will be formed. So the content of the DNA will double, but the number of chromosomes remain same. After the S phase, it is must for a cell to divide because the cell cannot have double the content of DNA in it forever. So then it enters into G2 phase. G2 phase is the further preparation where it prepares itself for the synthesis of new cells. So when the new cells come, so it will require some type of cell organism, some cytoplasm, some other things, and all that all will be collected here. Then it enters into M phase. Now let us concentrate on M phase. So this is M phase. What is M stand for? M stand for two things. M stand for mitosis or meiosis. So mitosis and meiosis are the two kinds of cell division. These are the two kinds of cell division. Mitosis is also known as equational division. And it is known as reductional division. Now, what is the basis of these equational and reductional names? These are actually in the equational, the two cells are formed are exactly alike. They have the same number of chromosome, same number, same type of genetic content on them. But in reductional division or meiosis, the number of chromosomes are reduced to half. So suppose it had n chromosome, then the four cells produced each will have five chromosomes. Okay. Now you'll say 5 तो 20 होना चाहिए, but that is यहाँ पे भी तो ऐसे ही हो रहा है 10 था, तो now the two are produced, each will have 10. Okay, so mitosis in which the two cells are produced, they will have exactly same number of chromosomes as the parental cell had. But in meiosis, four cells are produced and each will have half the number of chromosomes than the parental cell. So what it is required for? It is required for production of gametes. As we know, the gametes are either sperm and eggs, and sperm will have n number of chromosomes, egg will have n number of chromosomes, then they fertilize this, and the number of chromosome reaches to the same constant number what it originally had. Say for example 5 years and 5 years, so here become 10, which was the original number of the parent cell. Okay, so they are producing gametes. So it takes place only and only in those cells which are responsible for formation of gametes, that is gonads. So gonads, in animals it is testes and ovaries, in plants it is ovaries and stamens, okay, pistil and stamen, whatever you say, okay. But in mitosis can occur in all kind of cell, any kind of cell, okay, that can occur in haploid or diploid or prokaryotic or eukaryotic or unicellular or multicellular, any kind of cell, irrespective of any difference. So mitosis can occur in any kind of cell. Meiosis can occur only and only in gonads which produce gametes. And in mitosis, the two cells produces which have exactly same number of chromosomes. Meiosis will produce four cells, but they will have half the number of chromosomes. Rest, their importance and all, we'll study later. So now our concentration is only this much. We'll go ahead with the major differences, or rather what processes goes in it. So mitosis is completing in two phases. First is division of nucleus, and then division of cytoplasm. And the division of nucleus completes in four phases. This is prophase, metaphase, anaphase, and telophase. So at the end of this, we'll get two cells. यहाँ एक सेल डालेंगे, यहाँ दो निकलेगी. Now one should always remember, it is not like that the parent cell will be there and two more will produce. When the new cells are produced, the parent cell loses its identity and two daughter cells are produced. Okay, or rather we can put in this way that one is the parental cell and one daughter cell. Okay, so whatever way you put, but it is not that the two new cells will produce and parent cell is also there. तो तीन नहीं होंगे, वो दो ही होंगे. Meiosis. This is one cell division. Okay, so here the meiosis will complete in two divisions. So this is known as meiosis 1 and this is known as meiosis 2. Meiosis 1 is also completed in karyokinesis followed by cytokinesis. And karyokinesis will again complete in four phases: prophase, metaphase, anaphase, and telophase. And again the same process will repeated here. So at the end of the first meiotic division, two daughter cells will be there and they will undergo the second division, so karyokinesis, which will complete in four phases: prophase 2, metaphase 2. I'll simply write P2, M2, A2, and T2, where the P is standing for prophase, M for metaphase, A for anaphase, and T for telophase, and then cytokinesis. Okay, so this started with one cell. Here we had two, and the two cells will now result the formation of two more cells from each. So two, and here again two. So at the end of the meiosis, we'll have four cells. So meiosis completes in two meiotic divisions: first meiotic division and second meiotic division. First meiotic division is called meiosis 1, which will have four phases, that is prophase 1, metaphase 1, anaphase 1, and telophase 1. And second meiotic division, where the prophase 2, metaphase 2, anaphase 2, and telophase 2. So this is the outline of cell division and cell cycle. Okay, so in the next video we shall study about mitosis. So I think so far you should not have any doubt. So the next video we'll study about mitosis and then about meiosis and so on. Thank you.